So it's my honor and privilege today to introduce Dr. Eric Duncavidge, who's a physician scientist with a clinical interest in hematopathology and molecular genetic pathology. And his current research focuses on the development of new sequencing-based cancer diagnostic methods to understand the molecular mechanisms of myelodysplastic syndrome. He is a professor in the Department of Pathology and Immunology at the Washington University. He's also director of the section of hematopathology and director of the section of molecular oncology. He's a medical director of Cofactor Genomics and chief medical officer of Genoscopy. I hope I pronounced that correctly. Eric started his training as an undergraduate at Vanderbilt University, where he got his BS cum laude with a major in physics and chemistry, and then went on to medical school at the University of Tennessee in Memphis. Then he went to Washington University in St. Louis, where he did his combined APCP residency training. And that was followed by a molecular pathology fellowship at WashU, and then a hematopathology fellowship in Utah, the ARIP laboratories. And following his training, he became an assistant professor in the Department of Pathology at Washington University in 2011. Incidentally, that's when I started residency training there, and that's where our paths overlapped. So on a personal note, I can attest that Eric has been an outstanding mentor in pathology to both myself and numerous trainees. He has sent several of us to numerous USCAP meetings and been primary author on numerous translational pathology research studies. As early faculty, he had a position conducting research under the mentorship of giants in the leukemia field, Matthew Walter and Tim Lay. He developed an ultrasensitive sequencing method for the detection of low frequency variants in MDS and AML, and those were useful in detecting minimal residual disease. And his research then was supported by NIH K-12 and the Spore Career Enhancement Program. And he quickly rose the ranks since being an assistant professor appointed about 10 years ago. And he is now a full professor in the Department of Pathology. He's been co-PI and co-investigator on several NIH and institutional grants. He's published very well. He has over 85 high-quality PubMed indexed publications, including 2 first author publications in New England Journal of Medicine. The most recent one, 2021, is the genome sequencing as an alternative to cytogenetic analysis in myeloid cancers, which I presume that we're going to be hearing about today. And incidentally, I just this morning saw that this paper was lauded in a news press release from the NCI entitled Whole Genome Sequencing Could Help Guide AML Treatment. 2018, he had another first author New England Journal paper. He's got a patent for Myeloseq, which is a targeted panel sequencing for MDS and AML. He's on the editorial board of a really good journal, the Journal of Molecular Diagnostics. He's won 11 institutional and national awards, several membership and professional societies. He's given over 60 grand round and national meeting presentation, and just kind of try to condense how great he is in that short amount of time. Again, I just want to thank him for his flexibility, as he was supposed to give a talk in person last year. And I ask that everyone join me in welcoming Dr. Duncavage to our department Path Presents seminar series. And if there's any questions during his talk, please type them into the chat box and I'll read through them at the end of the presentation. And with that, please take it away, Eric.
Thanks for that very kind introduction, PJ. Let me make sure I get this set up. Can you guys see the slides?
Yes.
Okay, perfect. So I'm Eric Duncavidge, and I want to talk about whole genome sequencing for clinical cancer diagnostics. So PJ actually mentioned a lot of these disclosures unrelated to what I'm going to talk about today. So just kind of a basic overview of what I'm going to talk about. I'm going to talk about the current molecular testing approaches for myeloid malignancies. Hopefully I'll make the case to you guys for clinical whole genome sequencing. And then I'll talk a little bit about our paper on the subject. Then our assay is called Chroma-Seq and its performance in 263 AML and MDS patients. And then towards the end, I'll just briefly talk about kind of where we see clinical whole genome sequencing going and some potential applications. Okay, so AML classification has changed a lot over the years, and it was originally based on morphology. And so the original FAB system that was led by John Bennett was based on the morphology of blasts. And it turns out that the original thought was this correlated with outcome, but subsequent studies showed that the morphology, in fact, didn't recapitulate patient outcomes. And it turns out that it's really the genetics of AML that recapitulates patient outcomes. And so cytogenetics and molecular testing has really become critically important to myeloid malignancies. And so, I mean, every AML case that comes through here, even every MDS case, we do cytogenetics, we do panel-based sequencing, one-off gene sequencing, we do FISH. So it's really become part of the standard of care for AML and MDS. And why is it important? And so for AML, there's still very few targeted therapies, and so the name of the game is determining patient risk. And so we use the cytogenetic abnormalities and molecular data basically to determine, is the patient favorable risk, and those patients presumably go on to consolidation chemotherapy after induction, or are they adverse risk, and are these patients that we need to consider for transplant? And the reason, obviously, we can't transplant everybody is the cause. If you look at mortality from the transplant itself, it's roughly 15%. So we don't want to transplant everybody and expose them to that risk. So we have to be judicious. And so if you look at the most recent WHO classification, there are several. And the WHO classification itself now is based on recurrent cytogenetic abnormalities and molecular findings. So this is just kind of an overview of the really important core genetic findings in AML. And so how do we generate this data today? So we typically do metaphase karyotyping. And this is a culture and expansion of live cells, followed by direct visualization of the chromosomes under a microscope. We also typically do fluorescence in situ hybridization, which allows us to detect specific targeted alterations, including copy number alterations and gene rearrangements. And this is again done by microscopy, but by fluorescent microscopy. And metaphase cytogenetics is really, the methods were initially invented largely by Janet Rowley in the early 1960s. And the methods themselves have really changed very little in the last 50 years. There have been some advances, some use of automation, but the basic, basic principles of culture and making the metaphase spreads is really the same as it has been for 50 years. So it's remarkable. It's one of the few things, that and like the H&E slide, that has really stood the test of time in pathology. One downside is it does require the culture of living cells, which makes it not amenable to like solid tumors that are in general formalin fixed. It also requires direct visualization of chromosomes. So it's not a very high resolution assay. Another downside is that it's in our lab, it takes about 2 weeks, so about 14 days. And I mentioned, one of the good things about karyotyping, I think why it's lived as long as it has, or why it's been so hard to replace, is it's one of the few unbiased technologies. It is really the original genome-wide assay. It'll find, you don't have to ask specific questions like you do with FISH and look at specific loci. In theory, karyotyping will tell you what's there without any preconceived idea. And FISH is also fairly sensitive, and so it can be sensitive down to a level of about 1 in 200 or 0.5%, depending on if you're doing metaphase FISH or interface FISH and how the probes are validated. On the flip side, karyotyping in particular is subject to high failure rates. So if you look at publication or published studies, roughly 20% of AMLs have failed karyotype, and that can be due to a variety of reasons. Also, the turnaround times can be long. I know some labs can turn them around in as little as 5 days. Here, we're 14 and 21 days for karyotypes, and I think that's probably common for most academic labs. Karyotypes, again, you're just looking at DNA under the microscope, so it's not always possible to resolve complex events. FISH allows you to resolve some more complex events, but FISH probes are fairly expensive. It's very time-consuming to count FISH. So in general, the studies, at least in the clinical setting, are limited to just a few probes. And these methods too are also very, very labor intensive. They all involve direct visualization and highly skilled technologists to do these, to interpret these metaphase spreads. And so if you look at how we do cytogenetic and molecular testing at WashU for hematologic malignancies, we do conventional cytogenetics on basically every bone marrow, every AML and MDS that comes in. We also do metaphase FISH for AML. We do PML RARA, we do RUNX1 in version 16, and then we'll sprinkle in some other probes, DEL5, DEL7 in particular, as needed. We also we have a 40 gene targeted NGS panel, Aml-Seq, so we run that on every case. And then we run the FLT3 ITD companion diagnostic. And that's so basically for every AML and MDS case, we're doing these diverse set of testing. And so ideally, there would be one test to rule them all, right? So one test that so we don't have to, wait, and we don't have to consolidate findings from lots of different testing. And from a heme path side, I mean, this is kind of a huge problem for us, because we have to, at the end of the day, we're trying to collate data from lots of different tests. Some of it's send out, some of it's internal, it comes out at different times. And we're trying to put that together to basically tell the oncologists, this is an AML, to classify it by the 2018 WHO guidelines and really determine the risk. So other things or other technologies that have been proposed really as an alternative to cytogenetics, chromosomal microarrays. And some institutions, these are used. We don't use them for AML and MDS here. A big plus is they can detect much smaller copy number alterations. The problem, I think, has really been that they can't detect balanced rearrangements. So really only copy number alterations, not translocation. So it can't really replace cytogenetics from that standpoint. Mate pair sequencing, this is something that the Mayo group launched. And this is, I mean, in theory, this does detect rearrangements. It detects CNAs, but only large CNAs. The workflow is kind of complicated, and it turns out that Illumina stopped selling the kits to make these. So I believe this test from Mayo has been discontinued. Rna-Seq obviously can detect rearrangements, can also detect gene expression. And the downside there is, RNA is kind of is labile. It's a complicated process, doesn't always work. And then if you do RNA, then you still have to do DNA to find gene mutations. And combinations of targeted DNA and RNA sequencing, and this is kind of like what Tempus or Foundation of Medicine does. These approaches work, but they're kind of complicated. They take a long time, fairly expensive, and they're still, at the end of the day, they're biased. They can't detect. It's not like cytogenetics, where it can, in theory, detect any structural abnormality. It can only detect the ones that it's querying. And there have been other assays that have been published. This is a paper that was published a couple of years ago. For a comprehensive, AML assay. And they claim it's a simple approach, but if you look at this, I mean, it's fairly complicated. I mean, you're doing 3 different assays, DNA plus RNA, 6 different analyses. It takes 5 days. I mean, this is something that would be incredibly labor intensive. And this is out of Germany. And so, this would be very difficult for us to pull off here at WashU. So we asked the question, well, can we just use whole genome sequencing for AML and MDS diagnostics? And this is myself and Dave Spencer. So this is an idea we had probably about 3 years ago over lunch. One day we're just eating in the break room, and we were talking, we were discussing why, how can we still do all these complicated tests? Why can't we just do a whole genome? Like, there's no reason in theory why it wouldn't work. And so, I mean, whole genome can detect single gene mutations. I mean, so that kind of crosses that off the list. It can easily detect copy number alterations. Even low pass whole genome can detect copy number alterations, and detect translocations as well. And so, it seems like it's ideally suited. It's also a single assay with a simple workflow. And so Dave and I, we thought this was a great idea. We wrote a grant, and it got triaged. And so the agency didn't think it was that good of an idea. And so we were undeterred though. And we wrote, I think 6 months later, we wrote another grant. And this one somehow got funded. And so we were kind of off to figure out, how we can use whole genome sequencing, and how that for AML and MDS, and how that data compares to current workflows, so cytogenetics and FISH. Whole genome is also getting cheaper. I'll talk a little bit about that. And then along the way, very early, we realized that we had really no idea how to interpret cytogenetic findings, especially when you get into these kind of esoteric things like composite karyotypes and sidelines and things like this. So We collaborated with Molly Schroeder, who's one of our cytogeneticists here, and she was incredibly useful or helpful in the study and helping us interpret the data and translate. The other thing we want to do is translate the whole genome data into cytogenetic nomenclature. And so she helped with that. So I'll give you kind of 4 myths about, whole genome sequencing that usually come up when you start talking about it for clinical use. And the first that always comes up is, well, whole genome is too expensive. And the first human genome cost was probably about $1 billion. I think it's actually hard to calculate exactly how much it costs. This is expensive. The first AML genome, or the first cancer genome actually, was sequenced right here at WashU. That cost about $1 million in 2006, 2008. So not that long ago. Fast forward to today, in 2021, our core lab charges about $760 for a 30x whole genome. It's not very expensive. And if you compare that, I mean, that's a huge orders of magnitude drop in price over the last 15 years or so. And so it's kind of unprecedented. There are very few other things in life that have fallen, that the cost has fallen that much. And this is the slide that everyone always uses to illustrate genome costs versus Moore's law. So the price of sequencing has just plunged. So it's cheap and it's getting cheaper. And at the same time, while the price of sequencing is going down, price of labor is actually going up. And so, one of the things that we think is that long-term, it's going to be any assay that uses more sequencing and less labor will eventually become cheaper, and it's going to become probably more, applicable in the clinical laboratory. Another, thing that I hear is, oh, whole genome is so complicated. It's actually not true. It's the simplest of all the technologies from a wet lab standpoint, and we can actually make a DNA library in about 2 hours, a whole genome library, and it's easily automated. We run it on our Hamilton, and you load it right on the Nova-Seq. And actually, we use the Nextera Flex, which is I think now called Illumina DNA Prep. It's a tagmentation, a bead-based tagmentation assay. You don't even technically have to normalize the assay or normalize the genomes. You can just put them on the sequencer. So there's very little QC. It's an easy kind of foolproof method. And so it takes roughly 2 to 4 hours to use this bead-based tagmentation approach, which is what we used in our study. If you compare that to targeted Rna-Seq, I mean, this is the picture from the protocol from Illumina. I mean, it takes like 3 days, and it's like we've done this in our lab, and I mean, it really does take 3 days, and it's 3 busy days, and it's incredibly labor intensive. And the third myth, I think, is that, oh, the analysis takes forever. It's complicated. It's really slow. And I think, and this was a paper that was published by Elaine Martis, who was the director of the Genome Institute here at WashU. And there's a opinion piece that she published 10 years ago, kind of questioning, we hit the $1,000 genome, is it going to still cost, is it going to be $1,000 genome and $100,000 analysis? And that was kind of the thought, like, it was, oh, sequencing prices are going to fall, but the analysis is still going to be so complicated. And that hasn't really panned out. And so it is actually fairly fast to analyze the whole genome now. I think the biggest advance has really been the use of field programmable gate arrays like the Illumina Dragon. And so we can align and analyze a whole, or we can align a 60x whole genome in less than an hour. And we can do the full analysis, including variant calling, in less than 3 hours. In our previous methods, we have this pipeline called SpeedSeq, because it was thought to be fast like 3 years ago, and it still takes about a day and a half to 3 days to align a whole genome, and then even sorting a whole genome takes a long time in variant calling. So looking at like at least a week. But the Illumina Dragon has really, I think, changed a lot in terms of analysis. And then another myth, I think, that is that if you're going to do a whole genome, that you have to use paired normal tissue, and most clinical assays are tumor only, at least cancer assays. And so, and this has been, like, I think, a huge roadblock, and it comes up, especially for structural variants, or it does come up for structural variants. And so for like SNVs and indels, we can use population databases to figure out what's a inherited variant and what's presumably a somatic variant without sequencing paired normal tissue. But until recently, for copy number alterations and translocations, that was pretty hard. They weren't very well cataloged. And so this paper came out this past summer. My wife is actually the first author. So we were, we kind of had advanced knowledge. And so this using these 17,000 high quality genomes that had well identified structural variants, we were able to create basically a database of common structural variation. And we use that to remove the either artifacts or these inherited structural variants. And inherited structural variants are actually fairly common. So the average individual has, I think, about 3 structural variants that affect coding region, and usually about 4,000 total structural variants per genome. So they're pretty common. So you got to be able to map. You got to be able to remove them from your analysis. Otherwise, you're going to end up with a lot of junk or a lot of non cancer findings. And so our Chroma-Seq assay it's a tumor-only analysis. It uses rapid whole genome sequencing prep with the commercial kit, the Illumina DNA prep. And like I said, we can go from blood or bone marrow to a library, sequencer ready library in about 2 to 4 hours. We target 60x coverage on the Nova-Seq. We try to hit less than $2,000 per case. I'll talk a little bit at the end about cost and how that factors in. And then really, we spent a lot of time developing a simplified whole genome analysis that really focuses on clinically relevant genomic events. So translocation detection, megabase scale CNV detection, and targeted gene level analysis. And I mentioned this, you can call very small copy number alterations with especially with high coverage whole genome sequencing. For this study, what we've basically done is we've turned down the sensitivity for CNV, so we're only calling minimum size of 5 megabase per copy number alteration event. And we do that to make the results comparable with Cytogenetics, which probably has a sensitivity of around 5 to 10 megabases. So this is just kind of an overview. I mentioned a lot of this before, but basically, so we do this tagmentation based library prep. We sequence on the Illumina Nova-Seq 6000 instrument, and then we do this automated analysis. For the paper I'll talk about, we actually did the analysis on the cloud. We've since purchased our own local Dragon, and so we're doing, for clinical cases now, we're moving the analysis in-house. But the cloud actually works pretty well, and you just pay per case. It's like, I think, $15 for a genome. It's not very expensive. And so this is just an example here. This is a PML RARA fusion that we picked up. This is just like IGV screenshots here, just to kind of give you a general idea. But basically, you see these soft clipped reads and discordant paired end reads. We actually use Manta, which is freely available as a structural variant caller, is freely available from Illumina, and we've coupled that with a lot of custom filtering and scripts and things like that. But Manta really does the work, and it works, the heavy lifting, and it works pretty well. And then copy number alterations. And again, this is kind of, I think this is a great example of how nice, if you've ever tried to deal with like exome or targeted sequencing data calling copy number alterations, it's kind of messy. Whole genome, since it's unbiased, the CNA calls are really clean. And we use iCore CNV to do this. It's also, freely available with, we use some, downstream scripts to clean it up a little bit. But I mean, it runs pretty well out of the box on high coverage whole genome. And this is an example here. This is an MDS case with a +8. Here's a complex cytogenetics case. And we had to come up with our own definition basically for complex cytogenetics by whole genome sequencing. And so we call complex cytogenetics if we see 3 or more copy number alteration events. And again, the minimum size we call is 5 megabases. So we've kind of limited the sensitivity. This is an example of the Chroma-Seq output. It comes out from our pipeline. It's basically a text file. And we have, the copy number alterations will appear at the top. If there's any known translocations. And so we have a list of, I think, 617 recurrent translocations in myeloid malignancies. And if it's one of those known rearrangements, it kind of bubbles to the top of the list. And then gene level variants, just like you'd see on any assay. And then we also report previously, unreported, so not in our set of 617 high confidence structural variants. And we go through all this filtering to remove artifacts and false positives and things like that, especially from the translocation events. And then, then we take this, basically we take the raw output then, and then we kind of pretty it up and make these nice reports. And so we have a Circos plot here that demonstrates basically all the findings. So gene mutations are on the outside edge, copy number alterations here, and the translocations kind of in the middle. This is one of the first cases we sequenced. It turned out to have a cryptic 721 RUNX1 fusion that was not detected by our cytogenetics. But we went back and did FISH, and we actually, it panned out. And this patient, it's predicted to be, it's very rare, predicted to be high risk, and it turns out that this patient relapsed very quickly. And so that's kind of the idea too, really, that we had was we wanted to take the whole genome and basically distill it down to like one page. And we've heard from our clinicians, from our oncologists, the feedback we always get is like, reports are too long, these are too complicated. And so, we wanted to kind of compress all the clinically relevant information into a single page report. And we've, largely done that. Occasionally they extend to the second page. And so I'll tell you next about, kind of the technical kind of comparison of Chroma-Seq to cytogenetic testing. So we used a series of 263 patients. These are both AML and MDS patients. We used both retrospective and prospective samples. And we used the prospective samples really to get at, how practical is this of a technology to implement in the clinical lab. And then we also looked at outcomes from the AML patients in particular to determine if we performed risk stratification using whole genome sequencing, if that was comparable to risk stratification using standard cytogenetics, using the same ELN or NCCN guidelines. And this is our patient cohort again. So we have retrospective cases and prospective cases. The retrospective cases, again, we sequenced first just to get an idea of the technical ability of the assay. We also, so most of these cases had cytogenetic testing or gene panel-based testing. We also sequenced a subset of cases that had unsuccessful cytogenetic analysis, and I'll kind of show you some data from that as well. 117 of the samples were prospective. Most of those were AMLs, some of them were MDSs. And we sequenced these, really to get an idea for, if we put this in practice, how well does it really work? Again, this is just kind of a breakdown of the patients. It's kind of what you'd expect, I think, from a reasonable, AML cohort collected over, a couple of years, in AML and MDS. And we have a few other cases that kind of slip through. It's part of the prospective cohort, a couple of ALLs that we sequenced. That, because the way it was set up, we basically oftentimes we sequenced before we knew what the diagnosis was for the prospective cases. And so this is the sensitivity. So, and this is using all cases, so both the retrospective and the prospective cohort. So we found 100% sensitivity for the identification of translocations. So, we detected 40 recurrent translocations. Were detected by Cytogenetics. 100% sensitivity for copy number alterations detected by Cytogenetics, and 91 total events. We detected obviously all of the critical translocations 15, 17, inversion 16, 821 by whole genome sequencing. 100% sensitivity to detect complex cytogenetics. And again, we define complex cytogenetics from whole genome sequencing as having 3 or more copy number alterations. So 100% sensitivity, again, compared to conventional cytogenetics. And then sensitivity compared to targeted sequencing. So we compared to our Aml-Seq assay, which is, we target over 500x, typically about 1,000x coverage. And this is compared to, roughly, 50x coverage whole genome. The sensitivity is actually pretty good. So overall, 89% sensitivity for gene mutations with a VAF greater than 5%. It's actually 95% sensitivity for mutations greater than 10%. And I'll talk a little bit about that. And not surprisingly, all of the variants that we missed, the SNV and indel variants that we missed were not all, but the vast majority were low allele frequency. So it's just due to sampling. And we did some experiments where we boost the coverage on the sequencing, on the whole genome sequencing, and not surprisingly, we can detect these. And you can see this is just in a density plot of the mean of the ones not detected, the mean VAF, about 14% versus the mean VAF of those detected. So not surprisingly, low VAF mutations are harder to detect by lower coverage sequencing. And this is just looking specifically, and a lot of those mutations were subclonal, and probably, they're not, genes that would affect classification by NCCN guidelines, at least in AML. And so this is just looking at, these are the genes that do affect risk classification by NCCN guidelines in AML. And you can see our detection sensitivity for the ones that, the most important ones are pretty high, particularly phosmin. I think we missed one case. It was actually at a low VAF, 5%, and it co-occurred along with these other presumably high risk mutations, ASXL1. By NCCN guidelines, this is our only case that resulted in a change in classification, risk classification by NCCN guidelines. So just NPM alone would be favorable risk. We didn't detect that. By our assay, we detected ASXL1, so they'd be unfavorable risk. It turns out this patient actually was, I believe, died within 4 months. So probably, wasn't, or low risk or favorable risk probably undersold the actual risk in that patient. Anyway, so our sensitivity here for these recurrent genes is recurrently mutated genes is pretty good. We can also detect FLT3 ITDs. For our study, though, when I talk about the data, we relied on the companion diagnostic and not the FLT3 ITD calling by whole genome, although it does work pretty well. And we did this because, basically, I mean, we do the companion diagnostic on every patient, so we don't anticipate that would change with whole genome. And then another big advantage of whole genome compared to cytogenetics is you can find things that weren't identified by cytogenetics. So the next thing we did is we looked at the things that were missed by cytogenetics. So we identified 13 new translocations. 100% of those we confirmed either by FISH, PCR, or by RNA sequencing, depending on what the breakpoints looked like. We also identified new CNAs in several patients. Some of these were impossible to confirm, so we ran arrays on these. We used FISH where probes were available, and then we tried to go back and with higher coverage sequencing. But they weren't. It wasn't possible to validate all those. But in total, 40 out of 235 patients, and these are the ones that of the cohort that had cytogenetic testing had new findings, so 17%. So that's a pretty high number. And these aren't small events. These are big, recurrent events. And here's an example. This is a new CNA detected by Chroma-Seq. So this is you can see this del9 here. A minus Y wasn't detected. We also detected the A21, the RUNX1 rearrangement there, but wasn't detected by Cytogenetics. And you can see these are fairly low abundance, 13%. And you can still, even from this plot, without looking at the calls, you can pretty easily call these things. The next thing we did was we looked at prospective cohort. If we wanted to know, can we generate data in a clinically relevant time frame? And so we were doing these whole genomes basically in real time. When the Cytogenetics lab got a sample, they would split it, send some of it to, and they would send a pellet to us, and we would we'd extract DNA and then run whole genome. You can see when we got started, it took a little bit longer. Our turnaround times for the library QC generation and sequencing were pretty good throughout the process. The analysis at the very beginning took kind of a long time. We had to restart it a few times, and it was slow going at first, but we improved dramatically over time. And then here you can actually see towards the end of the experiment, we switched from this is all S4 flow cells. So we actually had to batch a little bit. And then we switched to S1 flow cells towards the end of the project. And that allowed us to basically do dedicated runs just for these for these cases. And that sped things up. And overall, our median processing time was 5.1 days. The shortest time we could turn around data was about 3 days. And also, of the 117 prospective patients, 110 of those, the reports basically came out and they were, they agreed. And there were only, 7 cases where we had to actually look at the CNV calls manually to try to figure out, to try to adjudicate the calls, in the report. And those were all low level events. And so we do have to look at plots occasionally. And looking at the diagnostic yield, and here, these are the prospective cases again. Here, we've just kind of honed in on just the AML patients. We did that because it's easier to calculate risk than it is for the MDS patients. And so overall, 25% of patients, AML patients, had new findings in this cohort. And 14%, so 10 of 68, those new findings would change the risk classification using the ELN risk category. So that's a pretty big, 15% of patients have a change in risk status. And I'll show you the Kaplan-Meiers from those in a second. And so what are those findings? And so 5 cryptic chromosomal rearrangements that we picked up out of these 68 cases. Almost all of those were KMT2A or MLL rearrangements, including in 2 patients there were two patients, they were poor risk MLL rearrangements. We picked up 1 721 rearrangement. We picked up 4 cases that had new CNAs that led us to classify them as complex cytogenetics, and then 8 cases that had incomplete or unsuccessful cytogenetics in that cohort, where we couldn't assign a definitive ELN or NCCN risk group. And these are the ELN NCCN guidelines that we used for risk stratification. And so, and this is looking at, our cohort of AML patients. here, 71 patients. And so this is just looking at applying the NCCN risk guidelines to these AML patients. And these are all, I should mention, these are non-transplanted patients. And so we remove the transplant patients from the list. And just applying NCCN to the conventional testing, so cytogenetics, FISH, and this is what it looks like. So favorable, intermediate, adverse, and then this is looking at the same patients, and now their risk is classified using the Chroma-Seq assay, so just using whole genome sequencing plus the FLT3 ITD PCR. And you can see we can largely recapitulate that. The intermediate risk patients actually kind of merge, actually kind of spread apart. Most of them actually go to adverse risk, and largely that's driven by the 4 patients that had that we called complex cytogenetics on by whole genome sequencing. But it does actually, so it does recapitulate outcomes using the standard ELN or NCCN guidelines. It's not we didn't come up with a new system for risk stratification AML. We're just using the same data. We're using whole genome data using in the same risk classification system that we've used for like the last 20 years or so. The other thing we wanted to look at is to see if we could predict clinical outcomes when cytogenetics were unsuccessful. So I mentioned before, roughly 20% of AML patients cytogenetics fails. And so can you use whole genome sequencing in this case? And so we picked a series of 27 AML patients where the cytogenetic risk was unknown because for various reasons, because there's no cytogenetic data, cytogenetics failed. And this is the outcome data. These are all non-transplanted patients looking at the outcome. And then so looking at now the outcomes based on sequencing. So we are able to actually, even in the small cohort, risk stratify. And so we can separate out the adverse from the favorable or intermediate patients. So we do see a significant difference there. So I think the immediate use for this assay is going to be for patients where conventional cytogenetics fails. Because again, it runs on DNA. I mean, we can run it on even if the the metaphase cultures fail, we can still run whole genome. And so just a brief summary of the Chroma-Seq study. So it's streamlined whole genome workflow. Hopefully I showed you data to indicate that it's practical, accurate, accessible, very simple to implement in the lab. The analysis is fairly rapid. Performance is excellent compared to standard cytogenetics. And again, we didn't design this to be a discovery assay. This is really designed to detect clinically relevant recurrent genomic events. The Chroma-Seq results from our study do recapitulate outcomes in AML using the standard genomic risk groups. And so you don't need to come up with a new system for risk classification. You can just use the existing classification system. And it's also a cost-effective approach to genomics. It could eventually replace the kind of fragmented testing we do today. And so comparison of prices and lab tests is always hard because it depends on like what you're talking about, costs or reimbursement, and if you're talking about reimbursement, who's paying. And so we go through, if you read our paper in the supplement, we go through various iterations of what the cost is. But I mean, the easiest thing to look at for standard testing is look at, well, what does CMS pay? And so this is for an AML workup at WashU. This is basically the published CMS rates. It doesn't necessarily mean it costs our lab that. It probably costs our lab more than that. And this is just kind of the lowest reimbursement you can expect, but still a fair amount of money when you add up all the testing and including FISH and cytogenetics and then panel-based testing. And then for Chroma-Seq, this is what we assume is the average cost to make a library here, including the labor, DNA quantification, DNA extraction, things like that. So about $273, and the cost of sequencing, and I'll talk a little bit more about that, but it ranges. So sequencing probably about $900 to $2,200 a case, and so you're looking at a total cost of anywhere from like $1,200 to $2,600, which is already kind of in the ballpark of what we're currently paying. So it's not. So the idea that this is something that's like outrageously expensive compared to conventional testing is not quite true. And again, I should mention too that it will be cheaper over time. And we looked at that, kind of that curve, the Moore's law curve. I mean, it's expensive today. It's going to be less expensive tomorrow. And I guess the other thing I should mention too is that if you look at the bulk of the costs here, it's in sequencing, right? So the labor, the reagents, something like that, it's a small fraction of the total cost. The big cost is the sequencing. And so if you compare that to our like targeted sequencing panels, roughly 80% of the cost of our targeted sequencing panels is labor or related to labor. Sequencing is a small fraction of that. And so, over time, any assay that uses, the cost is more weighted towards sequencing should fall because the sequencing prices are going to fall, whereas these other prices would go up. And so, kind of we're guessing that over time, the whole genome sequencing will become even more attractive to laboratories. And so, what does it cost to sequence a case? That's kind of a hard question because it depends on, how many cases do you have? And so Dave Spencer did this, really fancy simulation where we took into account, the different costs of the Illumina flow cells on the NovaSeq instrument, and then we modeled batch sizes and used this kind of fancy Monte Carlo simulation to figure out, to model like every week different potential batch sizes. And so, so we estimate, here, so we anticipate our in-house volume is somewhere around 200 cases a year. And so we're running this, we're going to run, we're actually the assay is actually validated now. We're going to start running it, I believe probably by May, we'll run it, clinically. And so, so we're anticipating about 200 cases a year internally. And that's just based on the we're going to run it on new diagnoses of AML and MDS, at least at first. And so, if you figure that out, you're running it on, you'd have to, based on those batch sizes, you'd have to run on S1 flow cells. That's going to be about $2,700 a case. But, if you can run on S4 flow cells with the new Illumina V1.5 pricing, it comes out to be much cheaper. And actually what our plan to do is we're going to run on S4 flow cells, even with 200 cases a year. And we're going to do that by, backfilling the flow cells with research cases, which has been our practice for the last few years. And so you can get the sequencing costs much lower, and you can see it scales pretty nicely. And so if you're doing 1,000 cases a year, 1,000, Chroma-Seq cases a year, the cost per case gets pretty low for sequencing. And this is again for 60x whole genome. And so finally, I was going to talk about, can you use this for other cancers? And so this is something we're very interested in, and we've spent a lot of time in the last few months. So lung adenocarcinoma is like a great example of it's probably the closest solid tumor type in terms of complex testing to hematologic malignancies. And so this is a lung adenocarcinoma case. We took a 1 millimeter punch of the FFP block. This is the actual area. It's pretty small, and for you guys that sign out lung cancers, it's not, not unheard of. And we just usually get like a couple of cores. And this is what the data looks like. We only got 48 nanograms of DNA, so not much. We got 84x coverage, 2.1 billion reads. So you can see this insert size is pretty small. Copy number plots, we didn't adjust the correlation matrix. And so it's a little bit noisy. And I think we probably do a lot better, but you can see, you can see these are believable copy number calls here. This is a case that we also sent to Tempus in parallel, and we were able to detect, and it had a kind of an odd NRG1 fusion. We were able to detect that from whole genome sequencing. Tempus detected it from RNA. We also detected a P53 variant. And so you can, use whole genome sequencing for, from FFP tissue. And we've done, a bunch of cases now, and it works. There are some caveats. And so how do we, what's kind of our future plan for this? How do we see whole genome sequencing fitting in with kind of the ecosystem of panel-based tests? And kind of our strategy, our kind of roadmap is, we envision that most cancer types, most solid tumors, will get a small panel up front, plus or minus FISH, depending on the cancer type. And that will really, determine frontline therapy decisions. And the feedback we've heard from our oncologists here is, we don't really need, 500 or 700 genes from some of these big commercial, panels to make treatment decisions. Like there's only a couple of genes that really make a difference in determining frontline therapy. And so, why spend money to sequence all these, all these genes on these patients that we probably don't need? And so then, and our plan is to use a tiered testing approach where, we would run the small panel plus or minus FISH, for example, on all lung cancer patients. If we find a mutation that allows the oncologist to set up, frontline therapy or choose a frontline therapy, then we're done. And if not, then we would reflex those to Chroma-Seq, and that would allow us to kind of like more judiciously use, whole genome sequencing. I think the idea of using it on every patient is probably a little premature, although you could. I think it's still kind of expensive. And for solid tumors in particular, there are issues with sensitivity due to stromal cell dilution. So this is kind of our plan, and we're working on implementing that now. And I think one of the major advantages of whole genome, as opposed to like these panel-based RNA plus DNA tests, is we can run whole genome from very small amounts of DNA. We can also run a small rapid panel from small amounts of DNA. So we can run these same assays from the same DNA extraction. So it's compatible with these small biopsy workflows. So that's how we're playing there. And with that, I'd just like to thank everyone that contributed. Obviously, Dave Spencer and Molly Schroeder, who helped with the study. The Cytogenomics laboratory here, the GAML PPGs, so Tim Lay, Sharon Heath, who helped with all the getting all the patient data information together. The oncologists who were key in helping us kind of interpret and guide us in terms of like what's clinically useful. And then Dave's lab and the people in my lab as well that helped with the analysis and all the samples and all the validation work that we had to do. So with that, I'll take any questions.
Well, thanks, Eric. That was great. While we wait to see if there's going to be some questions in the chat, again, thank you. Fantastic talk. Although I'm a little upset at the end that you used lung adenocarcinoma instead of glioblastoma as your example, but we can work through those issues.
Yeah, we've actually thought about that. I think that's whole genome sequencing on CNS tumors, I think is a great idea.
Yeah.
Because they're driven largely by translocations and copy number alterations. So it's something we've definitely thought of. We just haven't done it yet.
Yeah. And I think building off of some of our earlier work, that would be interesting to pursue, especially since we use it, our group, IHC, FISH, and then targeted sequencing. So I think this technology would be great for definitely reducing testing in our work environment. The first question I wanted to ask was, when you talked about the Chroma-Seq study that you published, you had mentioned that you're just really replacing the conventional testing and that you weren't actually using it for a discovery set, but you're just going along with current guidelines for risk-stratification. Have you mined through the data at all to look for, maybe use machine learning or some other applications to come up with improved risk-stratification?
We haven't done that. We've looked through the data, obviously, to find like obvious new things that were missed. I mean, there's nothing obvious. But yeah, that's a great idea. I mean, we're gearing up now to do sequence more cases, and I think we do anticipate. I think the last dark matter of the genome is really structural variants, and that's really the true power of whole genome sequencing. I mean, we're not going to find any new recurrent mutations, and to find a non-coding mutation that had some bearing on AML would be hard to find. You need a lot of patience. But I think potentially finding new structural variants is possible that are cytogenetically cryptic. We haven't really we found maybe one MDS case. It isn't in this cohort. It's in a different cohort that we didn't publish that has a potential cytogenetically cryptic driver event. But it's not. Yeah, I think it's going to be hard to discover new things, but there'll probably be a few structural variants, but I wouldn't expect them to be highly penetrant.
Okay, we have a question here from Rima Sorrentis. Asks, epigenetic changes are now key. How will make these HTS? I'm not sure what HTS means.
High throughput sequencing,
maybe.
Yeah, we're not doing. I mean, I don't know of anyone that does, epigenetics clinically. I mean, it's a huge focus. I mean, Dave Spencer's lab does. That's basically like all they do, and Tim Lay's lab does tons of epigenetic stuff. But it's still very much a research assay. We don't. I don't know of anyone doing epigenetic testing for hematologic malignancies in the clinical setting, and I don't think it would be hard to know like what it. Maybe 10 years from now, certainly once we understand like what it means, but it's still more of a research tool at this point.
Yeah, I was gonna just mention another for our brain tumor diagnostics. I think that's one that the looking at MGMT promoter methylation is a test that we do on everything, and it doesn't quite fit nicely within the standard NGS workflow. And that's another thing I was going to ask, if you could look at MGM or methylation and get it to be in the same workflow as this. Okay, we got one from Infang. It says the biggest pushback we got years ago when we rolled out chromosome genetic array testing was that risk stratification modification based on these new technologies are not necessarily the same as those defined by karyotype analysis based on large prospective clinical trials. For example, complex karyotype definition.
Yeah, no, that's good. And that's why we looked at the outcomes in these patients here to try to figure out, like, does whole genome based risk stratification correlate with risk stratification outcomes of patients based on standard cytogenetic testing. And it looks like it does. And again, you could always do more patients. We did a power calculation at some point to figure out, like, if we wanted to make it better, how many more patients we'd have to sequence. And it's like certainly way more than we could afford. And I think the next step really is for some of these big clinical trial cooperative groups to really test that. I mean, you would need thousands of patients to determine, is it really better at predicting outcome, or is it not as good as predicting outcome as the data generated by cytogenetics, or to come up with a new risk stratification system with a completely new definition of complex cytogenetics and things like that. I mean, that would have to be done by a big clinical trial group. I don't think any institution has enough patients either. These are still not common malignancies.
And has your group thought about incorporating this into like a biomarker-driven clinical trial?
Yeah, we have a couple clinical trials going on. The first one that we're going to do is with Megan Jacoby in the Division of Oncology. We're going to look to see how we're doing this. We just finished on Monday validating it in our CAP-CLIA lab here, the Chroma-Seq assay. And then we're waiting on signatures and things like that. And so we'll probably start issuing clinical reports in May, so next month. And we're going to look at we're going to look to see, like, does it we're going to look to see, we'll track the outcomes over time of these patients and look to but basically we'll do more prospective cases, and we'll see that we'll also survey the oncologists. We'll do this in parallel with cytogenetics, and we'll see, like, do our oncologists, will they act on the information, the whole genome information, or not?
I got one from Tina Lockwood.
Yeah.
Says, thanks for the great talk, Eric. Based on the 5% VAF limit of detection, what's your plan for clinical implementation? Diagnostic AML makes sense, but what is your plan for recurrence/monitoring?
Yeah, that's a great, so this is, we're not planning to use whole genome at every time point. And I think, we've already developed, assays for, high sensitivity MRD sequencing. Our thought is, like, we basically run Chroma-Seq on every new AML and MDS patient, or known patients at recurrence, but it's not sensitive enough to monitor. And so we would, in the interim time points, in the monitoring time points, and that's actually most marrows. I mean, an average AML patient generates probably 7 or 8 marrows over their lifetime. And so in most of those, we would run our high coverage MRD assay, Aml-Seq HD on. So that's kind of our plan with that. And I mean, my thought too, long term, as a hematopathologist, is, that eventually these MRD methods will probably, kind of supplant, like, a lot of these MRD methods and run on the blood. And that's the thing. Other thing we've figured out is blood and marrow, sequencing is largely equivalent. And so eventually that will supplant, like, looking at all these, surveillance bone marrow biopsies, like day 30, day 180, et cetera.
Another one from Minfang. According to your institution's current workflow, looks like RUNX1 and MLL translocations would have been picked up by existing standard of care at WashU. What is the net gain of detection rate compared with the combination of karyotype/FISH/array?
Yeah, and so we do RUNX1 on every patient FISH. We don't do MLL FISH, and that's another thing too with FISH. I mean, when we're trying to calculate cost and, like, things like that, I mean, every institution has their own, like, fish probe set that they run, and like we couldn't find any papers or any kind of national guidelines that say, like, you should run this set of fish probes. And so, we've chosen the ones that we do. Why MLL is not in that, I don't know. But and I can go back. Let's see. So what was the detection? It's here. That's the slide here. So, and so this is looking at the diagnostic yield. I guess it's not. So you're asking, if you include fish, we don't run arrays routinely. I think that's I don't have that. I don't have a slide with that. That's in our supplement. It's not because again, if you look at the ones that we missed, it's not far off from that because since we don't run MLL fish or KMT2A fish, these are not detected. This is not detected. The CNAs, wouldn't be detected by fish either. That led to complex cytogenetics. And obviously these I don't know, but it's probably comparable. So roughly, like slightly less than 25%. And again, not including and actually, yeah, you can see it on this table here. This these calls weren't detected. Most of these calls were not detected by cytogenetics or fish. So yeah, it would be, it's probably 20% or so, not including arrays.
Okay, I'm not seeing any more questions pop up yet. I think you addressed all mine.
Great. I think it's exciting. I think whole genome has, I think the next few years will probably be the, maybe the few years of whole genome. I think, it's still, I'd say it's still a little bit expensive. But I think, if the costs, again, like I mentioned, with the Illumina, with the 1.5 pricing, the cost dropped by like 50% this summer. If it drops again by another 50%, and what costs $1,100 is now like, $550. Like, I think that's going to drive, increased adoption. And I think, again, it's only, it should only get cheaper at this point. And so I think just the, that fact alone will drive a lot of, increased testing. And I think it's, at some point, we'll reach a limit where it's, it's just cheaper to sequence the whole genome and, throw away the parts that you don't want versus, doing some kind of upfront enrichment. Not quite there yet, but maybe in 10 years.
I did notice on one of your later plots when you were looking at the lung adenocarcinoma, you had those Manhattan copy number plots that obviously don't look as well as fresh tissue, right? Have you done head-to-head comparison of fresh versus FFPE to see what kind of noise there is in here and what you have to do for?
We haven't done paired testing. There's a couple papers out there that have looked at it. It's only like a handful of cases, maybe like 30 or something, if I recall correctly. Here we didn't. So the copy number alteration requires using like a matrix of normal samples to normalize to, and so we just ran this like cold using our pipeline for AML and MDS, and so it could probably get better if we'd actually fed it like a set of normal controls that were FFPE derived. And the big, and so that I think that's fixable. It is noisy, definitely more so than blood or marrow. The biggest challenge I think with whole genome from FFPE is going to be the insert size. So this insert size is, if you guys do a lot of sequencing, that's really small. And the problem is, to get 84x coverage, we had to do 2.1 billion reads, whereas we probably have to do half as many to get the same level of coverage if it were if the fragments were bigger, if the inserts were 300 base pairs or so, which is kind of like more on the end of fresh tissue. And so so the problem is that whole genome from formalin fixed tissue is more expensive to achieve the coverage depth you need than compared to fresh tissue, just because those inserts are really small. So we end up doing more sequencing to achieve the coverage depth that we want. So we're working. We've tinkered around with methods, for the last year or so to try to improve insert size, and we've made some progress. And so we're continuing to work on that to try to size selection and things like that to improve the performance.
Well, great. Thank you once again. 2:30. Everybody, I believe, thoroughly enjoyed the talk and has learned a lot. And again, we appreciate you taking the time to talk about your projects with us.